Okay, so I'm going to continue my lecture or resume my lecture on chapter 24. I'm going to start by just going over a couple of the chemical messengers that we spoke about last class and then pick up where we left off. So if you recall, we spoke about cholinergic messengers, and these are, this is predominantly acetylcholine, but it can also be any drug or a series of drugs that inhibit, enhance, or mimic the action of acetylcholine. We focused mostly on the acetylcholine receptors in skeletal muscles, and these are called nicotinic acetylcholine receptors because it causes an excitatory response at the neuromuscular junction in the skeletal muscle. So the ion channels open in response to the acetylcholine binding, and that causes depolarization of the target cell, and then the acetylcholine dumps into the channel. So you have those whole series of steps. Just go back and review that. But I did want to mention, and I didn't mention it the other day, is that it has the opposite effect in heart muscle. In heart muscle, it has an inhibitory effect. Now, there are two different types of acetylcholine protein receptors. So one receptor is found in the skeletal muscle and the other is found in the heart muscle. In the heart muscle, they don't open ion channels, but trigger signaling pathways in the target cell that inhibit the firing of an action potential. So they actually work opposite in two different locations. And I didn't mention the heart muscle the other day, which is important. Next, we talked about amino acid messengers. And we talked about glutamic acid, aspartic acid, and cytosine act as excitatory neurotransmitters, much in the same way as acetylcholine, epinephrine, norepinephrine, dopamine do. And then we talked about glycine, and then, which is an amino acid, and then two protein ones, beta alanine and gamma aminobutyric acid are inhibitory transmitters, and they reduce neurotransmission. Now, remember that glycine is the only one that actually is an amino acid. The other ones aren't. All of these guys have their own receptor, and they also involve a ligand-gated ion channel, similar to cholinergic receptors. And the last thing that I wanted to mention about that is that there isn't an enzyme usually that breaks down amino acid messengers, especially for glutamic acid. It has to be removed from the receptor site by a transporter molecule. Okay, so we left off talking about adrenergic messengers, and I started talking about monoamine messengers. And we got into the next slide, and that's where things got kind of confusing, and I was trying to make it simple for you. I don't know that I can make it simple, but I'm going to go through it again. We're going to start by saying that they transmit a signal very similar to the action of acetylcholine, meaning that they are absorbed on the receptor like the acetylcholine is. So once it's bound to the receptor, something has to happen to propagate a signal to the cell. And then that has to be transmitted throughout the cell. That whole process is called signal transduction. and it involves other compounds to help make that happen. In order to make that happen, you might have protein modifications, enzyme activation, and then the opening of membrane channels. There's a whole series of events that cause this to take place. So we're starting here, and you can see that the messenger neurotransmitter or hormone is adsorbed onto the receptor at the cell membrane, same way as we would acetylcholine. This receptor has an associated protein called the G protein. It has a long name, guanosine triphosphate. That protein is the key to propagating that signal throughout the cell. What makes it special is that it has or contains the nucleotide GTP, guanosine triphosphate, which is an analog for adenosine triphosphate. An analog is simply something that's similar to something else. So GTP is similar to ATP. The only difference is that you have guanine instead of adenine as the base. So this G protein, as you can see in the bottom part of the picture here, I'm going to see if I can circle it right here, hydrolyzes the GTP to GDP. When that happens, it activates adenylate cyclase, which is an enzyme. The adenylate cyclase cleaves the ATP into C-camp and something called pyrophosphate. This C-camp then activates another enzyme called protein kinase, which separates the regulatory unit from the catalytic unit, as you can see in the third picture right here. While this is happening, as you can see down in B, a second ATP phosphorylates the catalytic unit and produces ADP. When this happens, this is the last part, the catalytic unit phosphorylates the ion translocating protein, which is the last section that you see here, causing it to change its shape and position and opening the channel, which you actually see that down here. So even though it says A, B, and C, all of these things are happening at once. They're just trying to show you the different steps. And it's gone over in much more detail in the textbook, but I just want you to understand the process of how this happens and how these adrenergic messengers, especially the monoamines, work. So we talked about the G protein adenylate cyclase. That's the important part that gets this cascading of transduction signaling into motion. However, what's important to realize that these are not just limited to monoamine messengers. Glucagon, vasopressin, luteinizing hormone, and enkephalins, which bind to opiate receptors and are involved in levels of pain. Luteinizing hormones are involved in reproductive cycles. And honestly, I don't know what they mean by P protein, because there are like lots of different P proteins, and I couldn't find one specifically as it related to messaging, so I wouldn't worry about that one. A number of enzymes can be phosphorylated by the protein kinases, and the phosphorylation controls whether these enzymes will be active or inactive. So we talked about how this whole series of events produces the transduction across of the signal across the cell. But how do we remove that neurotransmitter or hormone? Well, it's the adenylate cyclase. That's the key in the removal. It stops the synthesis of the CCAMP or CAMP. And remember, the CAMP is what then, the cleavage of that is what causes the dissociation of the catalytic unit from the regulatory unit. The enzyme phosphodiesterase destroys the CAMP that is already produced. The phosphodiesterase causes the hydrolysis of one of the phosphodiester bonds, changing the CAMP back into AMP. This happens at a very, very slow rate, from 0.1 seconds to a few minutes. In cases where transmission must be fast, then a neurotransmitter such as acetylcholine has to act on the membrane permeability directly without a second messenger. Now, one thing that I want to make sure that you understand, you've heard me mention adenylate cyclase in two regards. So it has two important goals. The first is it can amplify the system in the beginning, okay? So once the neurotransmitter or hormone is absorbed onto the receptor, it triggers the adenylate cyclase to make many CAMP molecules. So that's propagating the signal. Now, in the removal of it, when the hormone or neurotransmitter dissociates itself from the receptor, the adenylate cyclase halts the manufacture of the CAMP. So it works in two different capacities depending on what's happening with that neurotransmitter or hormone. So now we're going to look at the series of reactions from A, B, and C, the original diagram that we were looking at. This is just showing you how the adenosine triphosphate is turned into CCAMP and pyrophosphate. It takes off two of the phosphorus groups in the triphosphate and leaving only one here and then the other two here. I don't expect you to know this reaction. I'm just explaining to you how it happens. So as long as it makes sense to you, what I'm talking about, then you're good. And then on the removal of the transmitter, it inactivates the monoamines by oxidation to an aldehyde. So you're taking that monoamine that has the amine group on it, the NH2, as you can see here and here, and converts it into an aldehyde. And it does that by an enzyme called monoamine oxidase. All right, so we're going to talk about histamines for a hot second here. The action of histamine as a neurotransmitter is very similar to that of other monoamines. So there's two kinds of receptors for histamine. One is the H1 histamine, which is found in the respiratory tract, where they affect vascular, muscular, and secretory changes associated with hay fever and asthma. Antihistamines that block H1 receptors relieve these systems. And then the other receptor is the H2 receptor, found mainly in the stomach, and that affects the secretion of HCl, cimetidine, and ranitidine, and they block H2 receptors and thus reduce acid secretion. A little trivia there, the main culprit in ulcers forming is a bacteria called Helicobacter pylori. And actually it was this guy, Sir James Black of the United Kingdom in 1988 received a Nobel Prize for the invention of cimetidine, which kills that bacteria. Just a little trivia. So we're moving on to peptidergic messengers, which are neurotransmitters that are made up of short peptide chains. The first brain peptides isolated were called the enkephalins. These pentapeptides are present in certain nerve cell terminals. They bind to specific pain receptors and seem to control pain perception. I mentioned that earlier, that they were involved in pain levels. In the last class, we were talking about mimicry. And here you can see that morphine, okay, has, or I should say, methionine and enkephalin has a end portion that is very similar or mimics that of morphine. Another brain peptide is called neuropeptide Y, and that affects the hypothalamus. So this has to do with your body's hormonal and nervous system. The neuropeptide Y is a potent orexic, which means appetite stimulating agent. When its receptors are blocked, then appetite is suppressed. The substance leptin, which is a protein, is considered an anorexic agent. So on a side note, there are some things that you can do to raise your leptin levels. One is getting 7 to 8 hours sleep a night, keeping some foods like apples, oatmeal, lean proteins, green tea, almonds, and eggs in your diet. Okay, so now we're going to talk a little bit about that substance P that they're referring to. P meaning for pain. This is an 11 amino acid peptide that's involved in the transmission of pain signals. So when you have an injury or an inflammation, your sensory nerves transmit pain signals from the peripheral nervous system to the spinal cord. So from where the injury occurred to your spinal cord, which processes the pain. The peripheral neurons synthesize and release substance P, which bonds to receptors on the surface of the spinal cord. And then this substance P removes the magnesium block at the NMDA receptor. And when this happens, glutamic acid, which we talked about earlier, which is your excitatory amino acid, can then bind to the receptor, and it amplifies the pain signal and sends a message to the brain. So we've been talking about various kinds of peptidergic messengers. And all of these peptidergic messengers affect metabolism. You know, we talked about a couple of specific ones, but others are insulin, glucagon, vasopressin, oxytocin, etc. But all of these peptidergic messengers act through a secondary messenger. As you probably already studied in bio, glucagon is a peptide hormone that is responsible for maintaining blood glucose levels. When the pancreas senses that your blood glucose level is dropping, it then releases glucagon. When it's released, it binds to receptors on liver cells, and then a series of reactions take place to raise the blood sugar. So you can see at A, you have glucagon bound to its receptor. Very similar events. What does it do? It activates the G protein, and the secondary messenger CAMP then activates protein kinase. We've been through this before, which is an enzyme that phosphorylates something else. So in this case, that something else is many different enzymes. So once the CAMP activates the protein kinase, a couple of things happen. You have the simultaneous phosphorylation of phosphofructokinase 2, which decreases activity, and the phosphorylation of fructose bisphosphate 2, which increases activity. These two phosphorylations of those two enzymes work collectively to produce a lower concentration of F2,6-P, which has a really long name, fructose 2,6-bisphosphate, which is just really referred to as F2,6-P, which is responsible for a lack of allosteric activation of phosphofructokinase 1 and the end result of lower glycolysis or increased gluconeogenesis. So, in short, what that means is increased gluconeogenesis means glucose is produced and glycolysis uses it. Also, by turning on gluconeogenesis and turning off glycolysis, the liver produces more glucose for the blood. Now, insulin is another peptide produced by the pancreas, but obviously you know its overall effect is going to be the opposite of glucagon. Insulin binds to insulin receptors on the liver and muscle cells, as seen here in this picture. What makes insulin receptor sites unique is that they have two different subunits, alpha and beta. The alpha subunit is on the extracellular side of the membrane, and it actually binds to the insulin, and the beta subunit spans the entire membrane. So when the insulin binds to the alpha, this causes the beta subunits to automatically phosphorylate the tyrosine residues on the other end. When the tyrosine subunits phosphorylate, this then causes the phosphorylation of insulin receptor substrates. These are just simply called IRS. Ha ha ha. And they act as a second messenger. All right, we are almost there. The last thing that we're going to talk about as a messenger are steroid messengers. These hormones are hydrophobic, meaning they are afraid of water, and they cross the plasma membranes by diffusion. Steroid hormones interact inside cells with protein receptors. Most of these hormones are ring structures, and they also include the sex hormones. They do not need any special receptors embedded in the membrane. They interact with protein receptors, and most of the protein receptors are in the nucleus of the cell. Some do, there are a small amount that exist in the cytoplasm though. Once they're inside the nucleus, the steroid receptor complex can either bind directly to DNA or combine with a transcription factor, which is a protein that binds to the DNA and then alters the expression of a gene. This then dictates which key proteins are synthesized. So steroid hormones can act in 3 specific ways. They can activate enzymes. 2, they can affect the gene transcription of an enzyme or protein. And lastly, they can change membrane permeability. I want to end with bringing this full circle and mentioning a few ways which drugs affect chemical communication, which is what this whole unit was about. A drug, whether it be pharmaceutical or recreational, can have effects on cellular communication, as you well know. It can affect the messenger, it can affect the receptor, the secondary messenger, or any one of the various enzymes that is activated or inhibited as part of a metabolic pathway, or that series of events that takes place to get the transduction signal rolling. An antagonist blocks the receptor and prevents the stimulation. An agonist competes with the natural messenger for the receptor site. And once it's there, it stimulates the receptor. Other drugs can work to decrease the concentration of the messenger by controlling the release of messengers from wherever they're being stored. And yet other drugs may increase the concentration of the messenger by inhibiting its removal from the receptor site. And lastly, some act to either inhibit or activate specific enzymes in the cell. I know this was a difficult chapter, but I will, in the review video, give you ideas of what to focus on. Okay, we are starting chapter 25 on Thursday.